Topic: the chemistry of life, organic compounds, proteins, enzymes, nucleic acids, vitamins. Aims of lesson. At the end of this lesson, you should be able to know the following. Proteins, amino acids, C, H, O, and N, and some have P, S, Fe. Proteins are sensitive to temperature and pH, loss of structure and function. Enzymes in breaking down/synthesizing molecules, influence of temperature and pH on enzyme. Action, lock and key model of how enzymes work. Enzymes in everyday life, e.g. washing. Powders. Nucleic acids, DNA and RNA, consisting of C, H, O, N, and P. No details of structure required. Of vitamins, e.g. A, one of B vitamins, C, D, and E. Introduction. Organic compounds all contain carbon. Organic compounds include food substances such as carbohydrates, lipids, proteins, enzymes, vitamins, and nucleic acids. Concepts and skills. Below are definitions of some of the terms that you will study under this section. Use the glossary in your textbook to define these new terms/words. Note: at least 2 marks are awarded if you can define a term correctly in a test or examination. Amino acids, monomers of proteins. Peptide link, bond between amino acids. Biuret test, the chemical test used to test for the presence of proteins. Know the meaning of instructional verbs in test and examination questions. Active site, the part of the enzyme that the substrate molecule fits into. Substrate, the substance which an enzyme acts upon. Anabolic reaction, type of reaction when a complex molecule is built up. Catabolic reaction, type of reaction when a complex molecule is broken down. Study the following information on proteins, enzymes, nucleic acids, and vitamins. Proteins. Proteins are large molecules, polymers, and are made up of the elements carbon, C, hydrogen, H, oxygen, O, and nitrogen, N. Some proteins also contain phosphorus, P, sulfur, S, and iron, Fe. Proteins are made up of long chains of amino acids which are the building blocks, monomers, of proteins. There are about 20 different amino acids. The sequence and type of amino acids determine the type of protein. The bond that forms between 2 amino acids is known as a peptide bond. A dipeptide is formed when 2 amino acids combine, and a tripeptide is formed when 3 amino acids combine. A polypeptide is a long chain of amino acids linked by peptide bonds. A protein consists of a chain of at least 50 and more amino acids. Answer the following questions. Question 1. 1. The following diagram represents apparatus used to investigate an organic nutrient. Study the diagram and answer the questions that follow. 2.1 Give the name of the nutrients tested for in this experiment. 2.2 State the color you would see if the result is positive. Proteins are sensitive to temperature and pH and will denature. Protein loses its shape and can no longer perform its function at high temperatures and extreme pH levels. Biological importance of proteins. Proteins are building materials, e.g. myosin in muscles, keratin in skin, hair and nails, etc. Proteins form a structural component of cell membranes. All enzymes and some hormones are proteins. Proteins are sources of energy. Chromosomes are made up of proteins and DNA. Test for proteins. A test for proteins. The Biuret test is used to test for proteins. Grind the test material, beans, and add a little water. Boil the test material until it forms a thin soup. Question 2. 2. The table below shows the nutritional value of a certain brand of breakfast cereal. Study the information before answering the questions below. 2.1 The total energy value of a 100g cereal is 2,000 kJ. The boy requires 500 kJ of energy per day. How many grams, g, of cereal does he need to eat in order to obtain this energy, assuming that he does not eat any other foods? Show your working. 2.2 Draw a pie chart to illustrate the proportions of protein, carbohydrate, fats, and fiber of this 100 g of cereal. Pour about 5 cubic centimeters of the soup into a test tube and add a few drops of dilute copper sulfate solution to the test tube. Add 5 cubic centimeters of sodium hydroxide solution to the mixture. Shake the test tube and heat the mixture. A violet, purple color indicates the presence of proteins. Enzymes. Enzymes are proteins. Enzymes are biological catalysts that speed up the rate of a chemical reaction without being used up in the chemical reaction itself. Chemical reactions that take place in living cells are known as metabolic reactions. Enzymes may be involved in two types of metabolic reactions, i.e. anabolic and catabolic reactions. Question 3. The diagram below shows the detail of an experiment done in a grade 10 class. 3 similar tablecloths, 1, 2, and 3, with identical blood stains, were washed in an enzyme-containing washing powder solution. The same washing machine was used at 3 different temperatures, 15 degrees Celsius, tablecloth 1, 37 degrees Celsius, tablecloth 2, and 65 degrees Celsius, tablecloth 3. 3.1 What is the purpose of this investigation? 3.2 Which property of enzymes is being investigated? 3.3 At what temperature did the enzyme operate the best/optimally? 3.4 Give a reason for your answer in question 3.3. 3.5 What could possibly have happened with the enzyme at 15 degrees Celsius? During an anabolic reaction, a complex molecule is built up from simple molecules. During a catabolic reaction, a complex reaction is broken down into simple molecules. During the digestion of food, complex molecules are broken down into smaller molecules. These reactions cannot take place without the help of enzymes. Lock and key model of enzyme action. The substance on which an enzyme acts is known as the substrate. The substance that is formed during the reaction is the product. Enzymes are substrate-specific, i.e. a specific enzyme can only act on a specific substrate, e.g. the enzyme sucrase will only react with sucrose, etc. The substrate fits into the enzyme's active site as a key fits into a lock and an enzyme-substrate complex is formed. The reaction takes place and the substrate becomes the product, which leaves the enzyme. The enzyme is now free to react with other substrate molecules. Question 4. Use the information in the graph below to answer the questions. For point 1, at which temperature does this enzyme work best? 4.2, how much food is broken down at 25 degrees Celsius? 4.3, explain the effect of temperature on enzyme activity. Enzymes are sensitive to changes in temperature. Most enzymes are inactive at low temperatures. The temperature at which enzymes work the best is known as their optimum temperature, e.g. 35 degrees Celsius, 40 degrees Celsius. An enzyme loses its shape and function and denatures at temperatures above the optimum temperature. Question 5. The following information, given in the table below, appeared on a box of breakfast cereal. 5.1 How many servings does a person need to provide the recommended daily allowance, RDA, of energy? 5.2 What deficiency disease could result if a person does not obtain adequate amounts of vitamin B1? 5.3 What deficiency disease could result if a person does not obtain adequate amounts of vitamin C? Effect of pH on enzyme activity. Enzymes are sensitive to changes in pH. Every enzyme has a pH range within which it can function. This is known as the optimum pH. Enzymes denature at extreme pH levels. Enzymes in everyday life. A biological washing powders contain enzymes e.g. lipase and protease that can break down stains. The enzyme protease breaks proteins down into smaller soluble molecules that can be removed by cleaning agents in the washing powder. Question 6. The following table shows an analysis of the nutrients found in a 100g portion of breakfast. The breakfast was made up of eggs, bread, and butter. 6.1 Name one nutrient that is essential for the manufacture of enzymes. 6.2 Identify the food type that would be best for a person who suffers from night blindness. Give a reason for your answer. 6.3 Calculate the amount of iron found in a 500g serving of the above breakfast. Show all calculations. 6.4 Which food type will show no color change when it is tested with biuret solution? 6.5 Briefly explain why an athlete should include bread in his/her breakfast. Nucleic acids. Nucleic acids are made up of the elements carbon, C, hydrogen, H, oxygen, O, nitrogen, N, and phosphorus, P. There are two types of nucleic acids, i.e. DNA, deoxyribonucleic acid, and RNA, ribonucleic acid. A DNA is found in the nucleus of a cell and it carries the hereditary characteristics. A RNA is found in the nucleus and cytoplasm of a cell and plays a role in protein synthesis. Vitamins. Vitamins are organic compounds and are required in small quantities by animals. Vitamins are essential for normal metabolism, growth, and development of the human body. Vitamins can be classified as water-soluble or fat-soluble. Water-soluble vitamins include vitamin C and the B vitamins. These vitamins are not stored in the body and need to be taken regularly. Fat-soluble vitamins include vitamins A, D, and E, which are stored in the body, e.g. the liver and fatty tissue. Question 7. Hydrogen peroxide, H2O2, is released as a byproduct during chemical reactions. The enzyme, catalase, converts hydrogen peroxide into water and oxygen. A grade 10 learner conducted an investigation to determine the effect of different temperatures on the action of the enzyme, catalase, found in raw chicken livers. Study the diagrams below. 7.1 In which of the test tubes, A, B, or C, will the reaction take place the fastest? 7.2. Give a reason for your answer in question 7.1. 7.3 Identify the independent variable in this investigation. 7.5 Identify the dependent variable in this investigation. 7.4 What would the result in test tube C have been if the chicken liver was cooked? Give a reason for your answer. Common errors made by learners in examinations. Unable to answer questions based on proteins, enzymes, nucleic acids, and vitamins. Unable to answer questions based on experiments to test for proteins. Unable to answer questions based on experiments of the effects of enzymes. Activities/assessment. Complete the questions given in this lesson. Consult your textbook, the internet, and other resources to find out more about the deficiency diseases night blindness, beriberi, scurvy, rickets, and anemia. Consolidation. Work through questions on organic compounds in past examination papers. Reflect on your learning and understanding of this topic and assess your progress. Values. By studying this section, you will develop your knowledge of key biological concepts, processes, systems, and theories.